Hi, I'm Vania Pardo, and I'm here with Aida Hernandez to talk about the BCR-ABL gene rearrangement in chronic myeloid leukemia. First of all, what is it? The BCR-ABL1 genetic rearrangement forms when parts of chromosome 9 and 22 separate and swap out. The broken piece of the chromosome 9 includes part of the ABL1 gene. It attaches to part of the BCR gene on chromosome 22. The modified chromosome 22 is known as the Philadelphia chromosome. You know why? Because of the city that where the researchers discovered it.
So now we have the research question. What is the differential impact of the BCR-ABL chromosome rearrangement on the progression of chronic myeloid leukemia compared to individuals without this genic alteration? Do you know, Vania?
Let's find out. Our hypothesis was that if individuals are diagnosed with chronic myeloid leukemia, then the frequency of the chromosomal rearrangement BCR-ABL is expected to be higher because BCR-ABL is exclusively found in individuals suffering from this type of leukemia.
So now we have the justification. The hypothesis is correct. The BCR-ABL gene associated with chronic myeloid leukemia, CML, is almost exclusively found in individuals with this disease.
For our results for the first experiment, we found that 14 out of 46 patients who presented the rearrangement BCR-ABL, and scientists developed a test called multiplex PCR to find this mix-up, and this test is accurate and can spot these gene mix-ups in many CML patients.
Okay, now, in the second experiment, scientists use a special test called multiplex PCR to check for specific gene changes in 45 patients with chromosomic myeloid leukemia. They look for a gene mix-up called BCR-ABL. In the test results, more of them, 97.77%, had a certain type of BCR-ABL mix-up. They also look at the patient chromosome under a microscope. More patients, 38 out of 45, had the standard BCR-ABL mix-up. Overall, in this study showed that multiplex PCR test was effective in finding a specific gene change in CML patients.
For the last experiment in this Indian population, more males had chronic myeloid leukemia than females. Younger patients often had one type of gene mix-up, B2A2, more than another type, B3A2. CML patients have different types of gene mix-ups depending on where their chromosomes break. Thank you.